Welcome to This Explainer. So today we're tackling organic chemistry, and we're totally demystifying it. We're going to take it from that terrifying wall of rote memorization and turn it into a highly predictable, kind of fun cast of recognizable personalities. Let's just dive right in.
Here's our roadmap for today. We'll hit molecule personalities, the neighborhood analogy, meet the chemical families, check out the carbonyl chameleon, look at one atom, two outcomes, and wrap up with the golden rule.
Section 1. Molecules have personalities. Reading the uniforms of chemistry.
Think about a basic carbon skeleton, like a molecule's, physical body. On its own, it's honestly pretty uneventful, just structurally stable and completely unreactive. But you know how you instantly recognize a person's job if they walk into a room wearing a chef's hat or a doctor's white coat? Chemistry works the exact same way. If you can spot a small cluster of atoms, what we call a functional group, it instantly tells you that molecule's chemical job. A tiny visual cue literally reveals the whole story. Spot an alcohol, that OH group, and boom, you know it dissolves in water. Spot an amine, an NH2 group, and you know it acts as a base. You're essentially just reading their chemical uniforms.
Section 2. The neighborhood analogy. Who lives inside?
To really grasp how powerful these functional groups are, picture carbon chains as identical houses in a suburban neighborhood. From the outside, the framework looks exactly the same, right? But what happens inside depends entirely on who actually lives there. Take house 1, ethane. It's just a simple, unreactive fuel. But if we swap out just one group, like moving a new family in, we get house 2, which is water-soluble ethanol. Swap it out one more time, and we've got house 3, acetic acid, that really sour stuff found in vinegar. It's the exact same carbon backbone every single time, but a completely different chemical personality.
Moving right along to Section 3, meet the chemical families, the oxygen and nitrogen groups.
So now that we've got a feel for how these chemical neighborhoods work, let's take a quick tour and meet a few of the locals. We're going to start with the perfume makers of organic chemistry, the esters. Formed by combining a carboxylic acid and an alcohol, these guys are less polar and have way lower boiling points than acids. What does that mean for us? Well, it means they easily vaporize. They're these highly volatile molecules that float right up and interact directly with the odor receptors in our noses.
And it's crazy to see the sheer variety of scents you can get from just tweaking that ester structure. Take isoamyl acetate. That gives us a really distinct banana aroma. But if you just swap out a couple of carbons to make ethyl butyrate, suddenly the whole thing smells exactly like pineapple. Add a ring structure to make methyl salicylate, and boom, you've got wintergreen. They all share that exact same core ester uniform, but those slight variations give them incredibly unique, specific aromatic personalities.
You can actually lock this into your brain with a super simple, punchy memory trick. Esters smell exciting. It perfectly reminds you of their pleasant, fruity vibe. It's just a great little mnemonic to keep in your back pocket for whenever you run into that COOR grouping.
Okay, now let's see what happens when we invite nitrogen to the party. Here we meet two heavy-hitting personalities. First up, amines. Think of them as the basic thinkers. Because nitrogen has a lone pair of electrons, it absolutely loves to accept protons, making amines basic. In fact, they show up heavily in your brain's neurotransmitters, like dopamine. Then we have amides, the protein builders. These guys form incredibly stable peptide bonds. They literally act like the glue holding your proteins and honestly, basically all of biology tightly together.
Section 4. The carbonyl chameleon. One bond, many identities.
Let's look at a structure that is so wildly versatile, it basically acts as the parent to 5 completely different functional groups. Ask yourself, what happens when one single chemical bond wears 5 different hats? The carbonyl group, which is literally just a single carbon atom double bonded to an oxygen atom, is a total chemical chameleon. It completely shifts its identity and behavior based entirely on who's sitting next to it.
So check this out. If that carbonyl sits right at the end of a carbon chain, bonded to a hydrogen, it's an aldehyde. But if it's tucked safely in the middle, flanked by 2 carbons, it's a ketone. Now, attach an OH group to it, and it instantly transforms into a sour, proton-donating carboxylic acid. Bonded to another oxygen chain, bam, it becomes our fruity ester. And if you attach it to a nitrogen, well, then it's that highly stable amide we were just talking about.
Everything branches out from that one central C double bond O. The big idea here is honestly wonderfully simple. Recognize the carbonyl group first, and then just look at its immediate neighbors to figure out its unique identity. It's the exact same core bond, but it yields completely different chemical personalities.
Section 5. One atom, two outcomes. The pharmacist's perspective.
Now, all this categorization is fascinating on a theoretical level, sure, but let's talk about the real-world stakes here, because they are incredibly high. Why do you think pharmacists obsess over such tiny structural changes? It's because they don't just memorize a giant list of drug names. No, they read the functional groups. They know that these little atomic uniforms entirely dictate a drug's safety, its efficacy, and its potential toxicity once it gets into the super complex environment of the human body.
Just look at the staggering difference between ethanol and methanol. Ethanol has 2 carbons attached to its OH group. It's safely metabolized by the body. I mean, it's the alcohol in celebratory drinks. But if you remove one single carbon atom to create methanol, it suddenly becomes a highly toxic poison. In the body, it metabolizes into formaldehyde, causing metabolic acidosis, blindness, and yeah, even death.
So the absolutely crucial takeaway here is that the difference between a pretty safe biological effect and lethal toxicity can literally come down to just one atom, one single carbon. This stark comparison really reinforces the most important rule in all of biology and pharmacology. Molecular structure entirely dictates biological function. Tweak a single functional block, and you can transform a nutrient right into a toxin or turn an inactive powder into a life-saving medicine. It's wild.
All right, final section, Section 6, the golden rule, thinking like a chemist.
We've seen the families, we've met the chameleons, and we know the serious real-world stakes. So how do we actually pull this all together into a practical strategy? Well, whenever you face an unfamiliar molecule, you've got to resist the urge to just blindly memorize its shape. Instead, use this really simple 2-step algorithm. Step 1. Identify the functional group. Just look for that chemical uniform. Step 2. Predict its consequences. Based entirely on that group, you can instantly predict its polarity, its acidity, solubility, and reactivity. The whole process is just elegant. Identify, predict, understand, apply. Once you confidently identify an OH group and immediately predict that it loves water, or you spot a COOH group and know instinctively that it donates protons, well, everything changes. You're no longer just staring at abstract letters and lines on a page. You are officially reading molecules like a professional chemist.
If you were to strip these functional groups away, the molecules of life would literally just stop folding, bonding, and reacting. Biology would grind to an absolute halt. Which leaves us with a fascinating question to ponder. If functional groups completely govern a single molecule's personality, how does your entire molecular structure dictate the very function of your life? What other incredible chemical personalities are hiding inside the biology all around us, just waiting to be read?